Dear students, today let us learn about semiclonal variation. Let us start with the introduction. The variations observed in the plants regenerated from any form of cell or tissues cultured in vitro is termed as semiclonal variation. The term semiclonal variation was first introduced by Larkin and Scowcroft in 1981 to describe all types of variations in plants derived from cells or tissue cultures. Semiclonal variations include genetic variability and variations for both qualitative and quantitative traits, and the plants regenerated from cell and tissue cultures are termed as semiclones, while the plants displaying variation are termed as semiclonal variants. According to Chalif 1981, plants regenerated from tissue cultures are labeled as R0 generation and their successive sexual generations as R1, R2 and so on. The type and frequency of semiclonal variations depends on several factors including explant source, ploidy and genotype of explant, age of culture, type of culture and pattern of organogenesis, culture media and physical conditions, etc. Generally, the chances of generating semiclonal variation is greater when ploidy levels of cells are lower than the usual ploidy level of the respective species and cultures are maintained for longer duration in callus and cell suspension phase of culture. Semiclonal variations can be divided into the following types based on the tissue from which variations originate. The first type of variation is gametoclonal variation or variations observed among the plants regenerated from gametic cultures. The second type of variations are the protoclonal variation or variation observed among the plants regenerated from protoplast cultures. The third type of variations are caliclonal variation or variation observed among the plants regenerated from callus cultures. Let us now see the sources of semiclonal variation. The semiclonal variations observed in the plants regenerated from cultured cells or tissues are derived broadly from two sources. The first one is the pre-existing variation in the somatic cells of the explants. Cells of meristematic explant such as apical meristem derived from either pseudapex or axillary bud divide by normal mitosis and produce plants with lesser degree of genetic variability. However, plants regenerated from non-meristematic explants generally produce genetic variability since the cells in these explants do not divide by normal mitosis but undergo DNA duplication and endoreduplication, and the cells may exhibit changes in chromosome numbers such as aneuploids and polyploids. The second source of variation is variation generated during tissue culture, induced by culture conditions. The plant cells undergo genetic and epigenetic changes induced by culture conditions. Generally, more variations are observed in plants regenerated from callus tissue compared to plants regenerated from other explants since callus contain mixed population of cells with different ploidy levels. Next, let us take up molecular basis of somaclonal variation. At molecular level, somaclonal variation may arise due to any of the following events. The first one is due to changes in chromosome number and or structure. Somaclonal variation may occur due to chromosomal abnormality in cultured cells, which may be due to changes in chromosome number or ploidy level and chromosomal structure. The second source is gene mutation. Somaclonal variation may involve recessive mutations of single gene in most cases, which do not show any apparent chirological changes and are inherited like traits showing Mendelian inheritance pattern. The third basis is plasmazine mutation. In vitro culture condition can also cause changes in plasmazines, which include plasmazine mutations and rearrangement in cytoplasmic genomes. Cytoplasmically inherited mutations have been observed in somaclones in several cases. Example, T-toxin resistance, male fertility, and mitochondrial DNA restriction pattern changes in TCMS maize. Alteration in gene expression. Somaclonal variation may also be due to alteration in gene expression resulting from the additive effects of mutated genes and changes in base sequences or activation of gene amplification. The fifth source is gene amplification. Some somaclonal variants recovered through stepwise selection of plant cells in vitro has also shown the occurrence of gene amplification. The next source is mitotic crossing over. Mitotic crossing over also account for some symmetric and asymmetric variation of the genetic variations detected in regenerated plants, leading to the recovery of homozygous recessive single gene mutations in the first generation of somaclones. Next is the transposable element activation. Somaclonal variation can also be caused by activation of transposable elements during in vitro culture. Next, let us learn about isolation of somaclonal variants. The various approaches to the isolation of somaclonal variants can be grouped into the following two broad categories. First one is screening and the second one is cell selection. Let us first learn about screening. It involves the observation of a large number of cells or regenerated plants for the detection of somaclonal variants. This approach is widely employed for the isolation of mutants for yield trait, and computer-based automated cell sorting devices have also been used to screen the desirable variant cells automatically. The next method is cell selection. It is the selection of somaclonal variants by preferential survival or growth of variant cells when a selection pressure is applied to the culture. Cell selection approach is called positive selection when the selection pressure allows only the mutant cells to survive or divide. On the other hand, it is called negative selection when a counter selection agent kills the wild type cells that divide normally and selects the mutant cells that escape the counter selection since they are unable to divide. The mutant cells are subsequently rescued by removal of the counter-selection agent, and it is a common approach for the isolation of auxotrophic mutants. The positive selection approach may be further subdivided into 4 categories. The first category is direct selection. In direct selection, toxins from pathogens, herbicides, elevated salt concentration, antibiotics, amino acid analogues, etc. are used as selection pressure to select mutant cells resistant to them, while the wild type cells are killed by the selection agent. The next is the rescue method. In the rescue method, the selection agent kills the wild type cells while the mutant cells remain alive but do not divide and are rescued by removing the selection agent. This approach has been used to select aluminum variant cells and variants resistant to low temperature, etc. Next is the stepwise selection. In this approach, the selection pressure, example salt concentration, may be gradually increased from a relatively low level to the cytotoxic level to select the resistant clones. The next type of method is the double selection. Double selection method uses selection for the presence of 2 features like survival and or growth as one feature and resistance to a selection pressure as the second feature. Let us now learn about the characterization of variants. Somaclonal variation can be characterized by using morphological, biochemical or DNA markers. Biochemical markers such as isozymes and DNA markers like random amplified polymorphic DNA or RAPDs, restriction fragment length polymorphism or RFLPs, and intersimple sequence repeats or ISSR, etc. are the most frequently used markers for the characterization of somaclonal variants. Coming to the advantages of somaclonal variation. The first advantage of somaclonal variation is that somaclonal variations have greater advantage over conventional mutagenesis, since somaclonal variations occur in higher frequencies compared to spontaneous mutation or chemically induced mutation. Secondly, somaclonal variations also serve as a source of new genotype that retain all the favorable characters of the existing cultivars while adding one additional trait, since new alleles or even new mutations may be isolated from different types of variants. Thirdly, the time required for the release of new variety is reduced with the use of somaclonal variation compared to mutation breeding, since somaclonal variants are usually free from undesirable features like sterility, which are generally associated with induced mutations. The fourth advantage is this approach is a highly efficient method, as effective selection can be practiced at the cell level for several traits, for example, disease resistance, etc., with relatively small effort, time, cost, and space requirements. The fifth advantage is it is the only approach for the isolation of biochemical mutants, especially auxotrophic mutants in plants. Let us now see the limitations of somaclonal variation. The technique is applicable only to those genotypes which regenerate complete plants in tissue culture. The selected cell lines may often show reduced or no regeneration potential and limit the application of somaclonal variations. The third limitation is many selected clones are unstable and show undesirable features like reduced fertility, growth and performance, and correlation between glasshouse and field performance of somaclonal variants is generally poor. The fourth limitation is it is a serious limitation for micropropagation, where the main aim is production of large number of uniform and true-to-type plants. The fifth limitation is somaclonal variants for most agronomic traits like yield and quality are not expressed and cannot be selected at the cell level and can be selected only by screening the progeny of tissue culture derived plants. The next limitation is many somaclonal variants may arise as the result of pleiotropic effects and may not be true variants. Let us now see the applications of somaclonal variations. First, somaclonal variations and gametoclonal variations are useful sources of new germplasm that could be introduced to plant breeders as valuable varieties. Since somaclonal variations can generate single gene mutation in nuclear or organelle genome, the variants may give rise to the best available variety that has a specific character. The third application is somaclonal variants of cell cultures can also be used as useful sources for commercial production of secondary metabolites, since variants of cell cultures have different potential for production of secondary metabolites. Fourthly, somaclonal variants for agronomically desirable traits like disease-resistant variants, stress-resistant mutants, mutants for efficient nutrient utilization, and other agronomic characters have been raised from tissue culture. Let us now see the conclusion. Somaclonal variation is an important method of genetic manipulation that can be used by plant breeders. The method has been effectively applied in certain crops with narrow genetic bases by providing a rapid source of variability for crop improvement. The chances of success for the application of the method depends upon the factors that affect the type and frequency of somaclonal variation like ploidy and genotype of explant, age of the culture, type of the culture, and pattern of organogenesis, culture media, and physical conditions, etc. Despite new improved varieties have been produced by somaclonal variation in a large number of cases, it is still not applicable in others since the selected cell lines showed reduced or no regeneration potential along with reduced fertility, growth, performance, and poor correlation between glasshouse and field performance of the somaclonal variants. Thank you.